Hello viewers, welcome to my channel. Today I will discuss about plasma membrane. So let's start the video. First of all, we will know about what is plasma membrane. Plasma membrane is the outermost boundary of cells and it is lipid bilayer, highly dynamic and fluid in structure, regulates the molecular trafficking, selectively permeable, that means not all the ions are enter into the plasma membrane, that's why it is called selectively permeable, electrically charged, that is inner part of plasma membrane is negatively charged and the outer layer is positively charged. Membrane potential is minus 20 megavolt and it is due to high potassium ion inside the cell. Here K plus ion is more, and it is amphipathic lipids, that means polar heads and non-polar tails are present. Polar heads are hydrophilic in nature and non-polar tails are hydrophobic in nature. Hydrophilic means water loving and hydrophobic means water fearing. Next, coming to the next slide, composition of plasma membrane. Plasma membrane consists of mainly three components. First is lipid, second proteins, and third is carbohydrates. Lipid is major part of the plasma membrane, 50%, and proteins also major part of the plasma membrane, it is 50%, and carbohydrate it is very few, that is 5 to 10%. Then lipid again divided into three components: phospholipid, glycolipid, and sterol. Then carbohydrate divided into two components. When carbohydrate mixed with proteins, it is called glycoproteins, and when carbohydrates mixed with lipids, it is called glycolipids. Then coming to the lipid bilayer composition and structural organization. The phospholipid bilayer, the basic structural unit of biomembranes. Each lipid have polar head and non-polar tail. This region is hydrophilic in nature and this region is hydrophobic in nature. Hydrophilic means this region is easily permeable to water, that means water loving region, and this region is water fearing, that means this region is impermeable to water and water soluble molecules and ions. Proteins embedded in the bilayer with specific functions. As bilayers, all biological membranes have an internal cytosolic part, phase, and external, that means exoplasmic. This region face to the cytoplasm and this region face outer of the cytoplasm. If we see, this is lipid molecule. If we enlarge, then I will see about this picture. Then coming to the next slide, proteins components. There are three types of proteins: structural proteins, carrier proteins, and enzymes. Structural proteins again divided into intrinsic or integral proteins, then extrinsic or peripheral proteins, and lipid anchored protein. Then catalytic protein, structural protein, transport protein. Transport protein again divided into carrier protein and channel protein. And many other more proteins are present in the plasma membrane, and each proteins have a specific function. If you require any type of query, then you may comment on the comment box. Then structure of plasma membrane, bilayer model. It is also called lipid membrane model. This model is postulated by Overton. In this model said that the plasma membrane is composed of thin layer of lipid. This layer is called lipid bilayer, and this is thin layer, and lipid soluble compounds crosses the, and this layer cross the plasma membrane does not, but in this model does not explain how ions are transported. Then Gorter and Grandel 1926 suggested that the plasma membrane composed of double layer of lipid molecules. The polar group of these molecules, this region, are situated on the outside of the layers. The Gorter and Grandel experiment on mature RBC, red blood cells, and he failed to explain how ion transport. Then next model, sandwich model. It is also called PLP, that is protein lipid protein model. In this model, Harvey and Cole 1931, Dabson and Danieli and Harvey 1935 indicated the presence of protein in the plasma membrane. In the previous model only said that lipid bilayer, but in this model, in addition to lipid bilayer, then it is also said that presence of protein. It is a trilaminar model like sandwich, and plasma membrane consisting of two layers of lipid molecules with their polar regions on the outer side. Globular proteins, these yellow color proteins are globular proteins thought to be associated with the polar groups. Lipids in the membrane consist mainly of phospholipid. This region is phospholipid with the non-polar groups near each and their polar heads directed outward. This region is outward and this is inner. Then unit membrane model. This model put forward by Robertson. The unit membrane is considered a biomolecular lipid layer between two protein layers. Here said that plasma membrane equal to bilayer. This bilayer lipid plus protein on both sides. This is inner protein layer and this is outer protein layer. Then most popular model is fluid mosaic model. This is widely accepted by Singer and Nicholson 1972. According to this model, the plasma membrane contains biomolecular lipid layer, both surface of which are interrupted by protein molecules. Protein occurs in the form of globular molecules. Here you can see the picture. Globular molecules are present and they are dotted about here and there in mosaic pattern. The extrinsic protein, that means outer protein, are attached at the polar surface, this region, and the lipid while integral protein, this is integral protein, are either partially penetrate the bilayer or span the membrane. This is span the membrane. On account of its fluidity and mosaic arrangement of protein, integral proteins are either partially molecules, and the model is known as fluid mosaic model. In the next slide, you can see the picture very clear. Here This is lipid and a polar head, and this is polar head, and this is lipid bilayer. You can see this protein is glycoprotein, carbohydrate present there. This glycolipid, this yellow is cholesterol. Integral, this is integral protein, that is all around the plasma membrane, and this is peripheral protein, only outside, and it is protein channel through which ion can enter into the plasma membrane. This is alpha helix span the plasma membrane, and phospholipid molecules are there, hydrophobic tails. This is hydrophilic heads. This is globular protein, surface protein, integral protein. You can see clearly. In the next slide you can also see the plasma membrane structure. You can pause the video and observe the picture very clearly. Then coming to the difference between integral protein and peripheral protein. What is integral protein and what is peripheral protein? Integral proteins are permanently attached to the membrane. This is integral protein. But in peripheral proteins are temporarily attached to the membrane, temporarily, not permanent. Embedded in whole membrane, located on the inner or outer surface of the phospholipid bilayer. Inner or outer part of the phospholipid bilayer. Then serve as integral protein, serve as carrier proteins, channels and enzymes, but peripheral proteins serve as receptor and surface antigens. Detergents should be used to remove integral proteins. Here peripheral proteins removed by salt pH changes. Glycoprotein are the example of integral proteins, and here erythrocyte spectrin are the example of peripheral proteins. Then fluid mosaic model versus sandwich model. Here you can separate them. Definition: fluid mosaic model. Fluid mosaic model is the model that states large protein molecules are embedded partially or completely within the lipid bilayer. But in case of sandwich model, the first model to describe the cell membrane structure stated that the phospholipid bilayer is sandwiched between two protein layers. Proteins are embedded either partially or completely, but in sandwich model there are two layers of proteins. Protein layers coat the outer surface only. Accuracy: the most accurate model and most accepted model, fluid mosaic model, but in sandwich model, first model, but not accurate. Penetration of proteins. Proteins permeate the lipid bilayer. Proteins do not permeate the lipid bilayer. Number of layers 2, but in case of sandwich model, number of layers 3. It is also called trilaminar model. Proposed by G.L. Nicholson and S.L. Singer, and sandwich model by Davison and James Danieli in 1935. Then last, coming to the function of plasma membrane. First function is protective. Forms of outermost boundary of the cells, that's why protect the inner organelles. Digestive, takes in food and excrete waste products. Selective permeability, help in transport across the membrane contains cell surface receptor, for example glycoprotein receptor present on red blood cells. Then cell adhesion molecules like cadherins present on the plasma membrane of certain cells plays an important role in process of inflammation, cell signaling, etc. And junction helps in information of various types of junction, that is adherence and anchoring, along with help of cytoskeleton elements. And many other more functions of plasma membrane are there. Some selective points. Then I am finishing my class here. If you like my video, then please like and share and comment and subscribe my channel. Click the bell icon to get new notification. Thank you.